We're moving on to all things relating to cells, and specifically to begin with eukaryotic cells. Now, cell theory states that all living things are composed of cells and that cells are the smallest components of life. The third point to make is that cells arise from pre-existing cells they cannot spontaneously generate. So what is a eukaryotic organism? A eukaryotic organism is made up of eukaryotic cells, and this means that those cells contain membrane-bound organelles. For example, the nucleus, the mitochondria, chloroplasts. This gives the compartmentalized cell structure that we're familiar with in animal, plant, and fungi cells. Now, organelles are the small components that you find inside your cells, and you'll have met lots of these at IGCSE and GCSE. So starting with the cell surface membrane, this controls the entry and exit of substances into and out of the cell. The nucleus, which previously you've learnt controls the activities of the cell, that's still true. It contains the cell's DNA, and the nucleus is surrounded by a double membrane which contains pores that allow substances to enter and leave the nucleus. There's a small subcompartment found within the nucleus known as the nucleolus, and this is the site of ribosome synthesis. Now, some ribosomes are found freely in the cytoplasm. These are known as ATS ribosomes, and they synthesize cytoplasmic proteins, so proteins found within the cytoplasm. Whereas other ribosomes are found located on the RER, the rough endoplasmic reticulum. These are used to synthesize proteins used outside the cell. Something new that you might not have met before is the centriole. This is used to assemble microtubules in the spindle for mitosis, and crucially, you do not find centrioles in plant cells. The cytoplasm you've met before. This is in effect the jelly of the cell, and it's the site of chemical reactions and the place where organelles are found. Now, the Golgi apparatus stores, modifies, and packages proteins. It often adds carbohydrates to those proteins in order to form glycoproteins. The SER stands for smooth endoplasmic reticulum. This is responsible for the synthesis of lipids. Lysosomes are small vesicles containing digestive enzymes which help to destroy damaged organelles and pathogens. And the mitochondria, again, this is the site of aerobic respiration and therefore ATP production. It means that energy can be released here. Remember, those plant cells contain the same set of organelles. They don't contain the centrioles, as I've already mentioned, but in addition they contain chloroplasts, which are the site of photosynthesis. They have a double membrane. The vacuole contains cell sap, and then you have the cell wall, which is made out of cellulose. This helps to support and maintain the shape of the cells. Fungi is another example of a eukaryote. Fungi include yeast, they include mucor. Now, what's different about fungal cells is that their cell walls are made out of chitin. Now we need to turn our attention to a group of definitions. So starting with a cell, this is a group of organelles which work together to perform a function. Going up a step, what about a tissue? This is a group of cells working together to perform a particular function. What about an organ system? It's a group of organs working together to perform a particular function. So the digestive system, for example, consists of many organs working together to perform the digestive functions, such as the stomach, esophagus, small intestine, pancreas, etc. Now, tissues become specialized in multicellular organisms, and that's so that they can carry out very specific functions. These tissues become specialized through a process of differentiation, and that's when certain genes are switched on within a genome and certain genes are switched off. Remember, the genome is the entire DNA belonging to an organism. So a very specialized cell type includes the sperm cell. Remember, the sperm cell's role is to fertilize the egg. It contains a haploid number of chromosomes in its head. That's so that when it meets the egg and they fuse, a zygote can be produced with a diploid number of chromosomes. But how is the sperm adapted for this function? Now, it has helical mitochondria which are packed into the midsection of the sperm. These produce the ATP needed to help the sperm swim. The acrosome is a special portion in the head of the sperm which contains digestive enzymes that help to penetrate the egg. Microtubules ensure that the sperm's tail beats side to side in order to propel that sperm forwards. After all, it's got a very long journey to make through the vagina, uterus, to the fallopian tubes, which is the site of fertilization. Protein fibers present in the tail help strengthen it, and we can also say that that tail is whiplash. Again, that helps to propel the sperm up that vagina to the uterus, to the fallopian tube. A second type of specialised cell, the red blood cell, at A level we call it the erythrocytes. They are adapted to transport as much oxygen as possible. How are they adapted? Well, they have no nucleus. This ensures that there's more space for the pigment hemoglobin. The hemoglobin binds to the oxygen and is a further adaptation. The red blood cells have a biconcave dish shape. This increases the surface area to volume ratio, again meaning that more oxygen can be transported. Last up, the erythrocytes are flexible, and that enables them to squeeze through narrow capillaries. Next up, neutrophils. These are a type of white blood cell. What's special about neutrophils is they have what's known as a multi-lobe nucleus, which enables them again to squeeze through small spaces. They have a granular cytoplasm which is full of lysozymes which help to digest pathogens using hydrolytic enzymes. So neutrophils are really an example of phagocytes, which you may be more familiar with when you were studying your IGCSE and GCSE. Moving on to different types of epithelial cells, the squamous epithelial cell. Squamous epithelial cells line blood vessels. They're smooth, flat and thin, and this provides a short diffusion pathway for substances. The ciliated epithelial cells, again you may have met these before, they line the trachea. They have small hairs known as cilia which waft backwards and forwards in order to remove mucus which is laden with dust and bacteria up your trachea to your mouth where it can be swallowed and destroyed by the hydrochloric acid. What about specific plant cells now? Remember, the palisade cell is full of chloroplasts. This is because we need photosynthesis to take place here in order to produce the glucose, the plant's food. What's also true about the palisade cell is that they have a large vacuole which helps to maintain turgor. That means that the plant cell remains nice and swollen. They are arranged vertically so they can be packed tightly and absorb as much of that light energy as possible. And they have thin walls to maximize the rate of diffusion of the carbon dioxide needed for photosynthesis. Root hair cells now, obviously located in the root, these have very large surface areas in order to absorb as much water by osmosis as possible in order to absorb mineral ions like nitrates through the process of active transport. Guard cells are located either side of the stomata in the leaf. The guard cells can swell up and become shriveled, and through doing that, they can control the opening and the closure of the stomata. Remember, the stomata is where transpiration occurs, so water loss from the leaf, and it also allows gas exchange. So the guard cells are very important for controlling the amount of carbon dioxide entering the leaf for photosynthesis, the amount of oxygen that's released in the process of photosynthesis. Obviously, if you're contemplating the plant's respiration, the gases will be moving in opposite directions. The structure of prokaryotic cells and viruses. So prokaryotic cells differ greatly from eukaryotic cells in that prokaryotic cells have no membrane-bound organelles. Bacteria are examples of prokaryotes. They display a simple cell structure with no compartmentalization of organelles. But what organelles do bacterial cells contain? They have a cell wall, which is made out of peptidoglycan, which supports and protects the cell. Bacterial cells have a capsule made out of carbohydrate that helps to prevent them from being engulfed. Their ribosomes are 70S. They're the site of protein synthesis. They have pili, which are hair-like structures that enable the transfer of information between cells. They have flagella, which are whip-like structures that help with the cell's motility. Rather than having a nucleus, they have what's known as a nucleoid, which is in effect a loop of DNA. And they finally have plasmids, which are small circles of DNA, and they actually help confer antibiotic resistance. So from a human point of view, the plasmids are pretty annoying. However, if you've studied genetic modification at GCSE, you'll understand the importance of plasmids in the manufacture of hormones such as insulin. We really need to unpick the difference between the genetic material found in eukaryotes and prokaryotes. As I've already mentioned, the DNA in bacterial cells, which are prokaryotes, is circular, so circular plasmids, whereas in eukaryotes it's linear. In eukaryotes, the DNA is associated with proteins known as histones. In prokaryotes, this DNA is naked. In eukaryotes, the DNA is found within a nucleus. In prokaryotes, it's found freely in the cytoplasm. Now we need to mention viruses. Now viruses are neither prokaryotes or eukaryotes. We consider them to be non-living. because they have no metabolism and they need another host cell in order to reproduce. They cannot reproduce by themselves. In terms of their structure, they contain genetic material, depending on the virus, that will be either DNA or RNA. They have a capsid which surrounds the virus particle. This is a protein coat which surrounds the genetic material. They have an envelope, and this is actually made out of the phospholipid bilayer present in the host cell. They have really annoying attachment proteins which are complementary to receptors on host cells and helps them penetrate the host cell. And they also contain enzymes such as reverse transcriptase, which is found in HIV. And this enzyme helps to incorporate the virus's RNA and DNA into the host's DNA. Bacteriophages are viruses that kill bacteria. The diagram below shows drawings of a bacteriophage and a bacterium. Using the diagram and your own knowledge, put a tick in the box next to the only correct statement about the structures of bacteriophage and bacteria. So there's your virus. Both have ribosomes. That's not true. Viruses don't contain ribosomes. Both have a cell surface membrane. Again, not true. The bacteriophage has a capsid, yes, and the bacteria has a cell surface membrane. Let's have a look. Is that true? Yeah. There's our capsid. Here's our cell surface membrane. Correct. The bacteriophage has a cell wall. No, it's a virus. It doesn't have a cell wall. The bacterium has a capsid. No, that is true of viruses. So the answer here is the third box. Below is a diagram of an animal cell. Do get used to recognizing your animal cells. You still have your characteristic fried egg shape with your nucleus here, your cell surface membrane. Obviously, we have the mitochondria. Here's your rough endoplasmic reticulum, characteristic due to the presence of those ribosomes. This is your Golgi body, which I always think looks a little bit like an amphitheatre, and here are your free ribosomes. Name the organelles labelled B. That's your Golgi apparatus. And then C is your mitochondria. Name 2 structures present in plant cells that are not present in animal cells. Not too difficult. Vacuole, chloroplasts. Complete the table to show the features of a bacterium and a virus. Put a tick in the box if the feature is shown. Bacterium has a cell surface membrane, viruses do not. Bacterium does not have a nucleus, it has a nucleoid. Neither do viruses. Bacteria has cytoplasm, virus does not. The virus has a capsid. DNA and RNA can be found in bacteria. Give 2 ways in which the nucleotides in DNA are different from the nucleotides in RNA. DNA contains thymine. RNA contains the base uracil. Let's name the differences in the sugar. DNA has deoxyribose, sugar, in its nucleotide, whereas RNA just contains ribose. Now we turn our attention to microscopes. We'll be looking at various keywords to do with resolution, magnification, how you set up a light microscope, as well as the major differences between light and electron microscopes. And with electron microscopes, we'll be comparing transmission and scanning. So the 2 main things we like to consider when choosing our microscope is both the magnification and the resolution of the particular microscope. So the magnification is how many times larger the image is compared with the original object. Resolution is all about the ability to distinguish between 2 separate points. So the higher the resolution, the clearer the image. We can do a calculation in order to determine magnification. Magnification is image size divided by actual size. So use a formula triangle I am, and that will enable you to rearrange that equation to find image size, magnification, and actual size, depending on the data they give you in the question. One thing to be aware of is you will need to make sure that your image size and actual size are in the same units. That's often micrometers. So make sure you're happy converting between millimeters to micrometers. You need to multiply by 1,000. Obviously do the opposite if you're going from micrometers to millimeters. The most straightforward simple microscope is your light or optical microscope. You've probably used this in a laboratory if you're at school. Simply put, a light microscope has the following components: an objective lens which focuses light and increases magnification, your eyepiece lens which further increases magnification, and your all-important light source, a lamp which shines light through your specimen so you can actually see it. Remember, when you're using your light microscope, you want to start at the lowest objective lens and then slowly work your way up, increasing the magnification. You may need to adjust the height of the stage in order to bring that slide into focus. In addition to those three components, you might have heard of an eyepiece graticule. Its purpose is to measure the size of an object when viewed with a microscope, and this must be calibrated every time the magnification is changed. The stage micrometer is used to calibrate the eyepiece graticule every time the magnification is changed. So simply put, how do we prepare a sample for a light microscope? Well, we need to mount it on a slide. So first of all, we need a thin slice of the sample. It needs to be thin so that light can shine through it and it allows the details of your sample to be seen. Next up, you need to suspend your sample in a liquid such as water or oil. Then place your cover slip over your sample. You need to place that at an angle to stop air bubbles being trapped, which would disrupt the image. And then finally, press down that cover slip using a lens tissue. You'll often find that your sample is squashed beforehand, and that's to ensure that there's a single layer. Other things we often do is add a stain to our sample. That enables us to distinguish between different structures within the cells, and differential staining simply means using multiple stains. Each stain binds to a different structure, so it makes it very easy to distinguish particular structures within a sample. Now, you'll often find if you're observing a plant cell that they'll use shoot tips. The reason for that is because the shoot tips, obviously they're growing really quickly, so they're undergoing mitosis, which is a type of cell division, and it enables us to see the various stages of mitosis really easily, such as prophase, metaphase, anaphase, or telophase. Now, in terms of light microscopes, their magnification ranges between 500 and 1,500 times bigger than the original object. Their resolving power is just 200 nanometers. The image type created is 2D, and the smallest structure you can observe with a light microscope is a mitochondria. So obviously the magnification resolving powers aren't great of light microscopes, so why do we still use them? Well, it's simply because they are comparatively cheap to buy, they are comparatively easy to use, and a big plus is that they can be used to observe both live and dead organisms. So you might have observed water fleas, Daphnia, down a light microscope, and they are very much still alive. Electron microscopes have much greater magnifying and resolving powers. There are two types, the transmission and scanning electron microscope. The scanning electron microscope has a resolving power of between 3 to 10 nanometers, so much better than the light microscope. They can magnify up to 100,000 times greater than the original object. Images created can be 3D, and the smallest structure you can observe with a scanning electron microscope, SEM, is the ribosome used in protein synthesis. Transmission electron microscopes are even better than scanning electron microscopes. Their resolving power is 0.5 nanometers, so they can distinguish between 2 points just 0.5 nanometers apart. They can magnify up to 500,000 times. One disadvantage is that images created are 2D, and the smallest object they can distinguish is the cilia on ciliated epithelial cells. So what's the major reason for electron microscopes having a much greater resolving power compared with light microscopes? Well, it's all to do with the wavelengths involved. The wavelength of light is much greater than the wavelength of an electron. One small detail to mention is artifacts. Now, these are objects which appear on your image due to the way in which the sample has been prepared. So they're not part of the natural specimen, and it's actually quite hard to work out when you're looking at the image if these artifacts are actual organelles or if they're just damage. A second method for studying cell structures is known as cell fractionation. This is a way of breaking up cells in order to be able to study individual structures. It can help us discern the specific functioning of organelles. Specific conditions are needed to prepare a sample for cell fractionation. First of all, we need cold conditions. That's to stop hydrolytic enzymes present in the sample from working and breaking up the structures. You need to bathe the sample in a solution with the same water potential as the original sample, and that's to prevent osmotic activity, because you don't want to draw water into and out of the sample. Because particularly if you've got an animal cell, as they're lacking a cell wall, any water moving in could cause the animal cell to burst. You also want to use a buffer solution, and that's to prevent changes in pH from damaging your sample. There are two stages of cell fractionation. The first stage is homogenation. The second stage is ultracentrifugation. That's a hard word to say. So how does homogenation work? Well, actually it sounds quite drastic, but you're going to use a blender to blitz your sample. That ensures that the organelles are released. You then place what's known as the homogenate through a filter, and that removes whole cells as well as any debris. Before ultracentrifugation can take place, you're going to treat your sample with a detergent, and that's so that the detergent can release any DNA and protein from the cells. The reason why detergents are used is because cell membranes consist of a phospholipid bilayer. The phospholipid bilayer is broken down using detergents. And that explains why detergents are great at removing fat and grease marks from our clothes. So what is ultracentrifugation? It's all to do with spinning the sample in order to separate out the contents. So with ultracentrifugation, we start at a low speed, and what happens is the heavier organelles, such as nuclei, sink towards the bottom of our test tube. And here they form a pellet. Fluid collects at the top of the tube. This fluid is known as supernatant, and this is transferred to another tube. Then this is placed back into the centrifuge and spun again, this time at a higher speed, and then the middling-sized organelles sink towards the bottom, including mitochondria. Again, the fluid collects at the top of the test tube, the supernatant. This is transferred onwards, and it's spun again, and by doing this we can separate all our organelles out. So by the time we reach the highest speeds, it's only the smallest organelles which are left, such as ribosomes. Uranema marinum is a single-celled eukaryotic organism. The diagram below is a photograph of U. marinum taken through an optical microscope, in other words, a light microscope. Explain why it's not possible to determine the identity of the structures labelled X using an optical microscope. Quite frankly, when you look at them, you can't distinguish between the different organelles, so that means that the resolving power of the light microscope is too low. And the reason for that is because the wavelength of light is too long. That lambda there refers to wavelength. U. marinum cells ingest bacteria and digest them in the cytoplasm. Describe the role of one named organelle in digesting these bacteria. Remember, these are lysosomes. They fuse with the vesicle and release hydrolytic enzymes. Calculate the actual length of the cell shown between Y and Z in the diagram above. The magnification of the image is times 900. Give your answer in micrometers into 2 sig fig. So I'm going to use my formula triangle. I'm after the actual length of the cell, so I'm going to do actual length equals image length divided by magnification. So what is the image length? You need to use your ruler in the exam to measure the length of this, which is 2.9 centimeters, which is 29 millimeters. The magnification, we were told, was times 900. Careful with your units, that's in millimeters. Times it by 1,000 to get it into micrometers. And then don't lose a mark, you need it to 2 sig fig. Give 2 structures found in all prokaryotic cells and all eukaryotic cells: cytoplasm, cell surface membrane. The figure below shows transmission electron micrographs of 2 cells, one animal cell and one prokaryotic cell. Contrast the structure of the 2 cells visible in the electron micrographs shown in the figure above. This is obviously our bacterial cell, the prokaryote. This is my animal cell. First of all, what can we see inside our animal cell? Well, we can see a nucleus, whereas here all we can see is a nucleoid. Here are some mitochondria surrounded by double membranes. They are absent in cell B. We've got a Golgi apparatus here. Again, that's absent in B. Cell B has both a capsule and a cell wall. Cell A has no capsule or cell wall. It simply has a cell surface membrane. This is present in cell B also. What about ribosomes? The ribosomes here are much smaller and they're 70S, whereas cell A has larger ribosomes. So cell A has a nucleus, mitochondria, Golgi apparatus, no cell wall, no capsule, no plasmids, so no circular DNA, larger ribosomes. Cell B has no nucleus, no mitochondria, no Golgi apparatus, but does have a cell wall, capsule, and plasmids. The image below is a transmission electron micrograph of a plant cell. Suggests why a nucleus is not visible in the above image. One thing I do want to point out is you do need to familiarise yourself with these various micrographs. Try and look at as many as possible, because your nice neat image of a plant cell like this that you're so familiar with, or your animal cell being like this, unfortunately it's not really the reality at all here, as you can see from this plant cell. However, you can distinguish the plant cell wall, the cell membrane. These are your green chloroplasts, your vacuole. It says suggests why a nucleus is not visible in the above image. It's not saying that it's absent, it's just saying why isn't it visible. So you could say anything like, oh, this particular slice didn't contain the nucleus, it was present elsewhere, or you could say that they haven't stained the nucleus. Name the organelles labelled S and T. I've already mentioned them. Here's your vacuole filled with cell sap. Here's your green chloroplast. Give one advantage of viewing a biological specimen using a transmission electron microscope compared with using a scanning electron microscope. So remember, we said that transmission electron microscopes can magnify up to 500,000 times, whereas scanning is comparatively only 100,000 times. Something else you could point out is the difference between their resolving power. Remember, we said that transition microscopes can do 0.5 nanometers, whereas the scanning electron microscope can, again, in inverted commas, only do 3 to 10 nanometers. So in my answer, I've pointed out that transition electron microscopes have a higher resolving power of 0.5 nanometers compared with the scanning electron microscopes 3 to 10. This is an interesting question because it really wants to check that you can sort out your units. So place a tick in the box next to the sequence that shows the correct order of magnitude of these measurements. So I've just written here a summary of unit conversions, so how to get from nanometers to micrometers, micrometers to millimeters, millimeters to meters. So let's do some unit conversion. I'm going to get them all into meters. So I'm going to do 50 divided by 1,000 3 times. So that's 5 times 10 to the minus 8 meters. What's 0.5 micrometers in meters? 5 times 10 to the minus 7. 5 times 10 to the minus 2 millimeters in meters. But then look, is 5 times 10 to the minus 5 meters smaller than 0.5 times 10 to the minus 5? Obviously not, so that means that one's wrong. Let's move on to this one. What's 50 nanometers in meters? So we know that this again is 5 times 10 to the minus 8. 5 times 10 to the minus 7. This is fine because we know that 0.5 times 10 to the minus 5 is larger than 5 times 10 to the minus 7. And then lastly, let's get 5 times 10 to the minus 2 in meters. So this is 5 times 10 to the minus 5 meters. So go through them. Yes, that's smaller than that. This is smaller than this, and this is smaller than this. So that's why B is the correct answer. Name an organelle found in both a chloroplast and a prokaryotic cell. So prokaryotic cell is something like a bacteria. The chloroplast, remember, is an organelle found within a plant cell. That makes it slightly trickier because it's asking what's found within a chloroplast. So your best option here is to pick ribosome, because after all, you need a smaller organelle than a chloroplast. A scientist determined the volume of a plant cell and the volume of organelles it contained. They found the volume of a plant cell is this. The volume of all the mitochondria in the plant cell is this. The volume of all the mitochondria and all the chloroplasts in a plant cell is 44.1% of the volume of the plant cell. Use this information to calculate the volume of all the chloroplasts in a plant cell. So first of all, we need to find out what 44.1% of the total volume of a plant cell is. And then we know what the volume of the mitochondria is, so we need to take that away. And that means that is the volume of chloroplast, because we were told that together the volume of the mitochondria plus the chloroplast was 44.1%. The diagram shows a section through a plant cell at magnification times 500. Calculate the actual diameter of the starch grain between points A and B. So here's the formula triangle I mentioned, IAM. We're being asked for actual, so it's image size divided by magnification. So do measure the length of that starch grain using your ruler. If you've measured that carefully, it's 20 millimeters. Just be a little bit careful with your units. The final answer is in micrometers. So to get it into micrometers, we need to times by 1,000. And then divided by the magnification, which we were told was 500. So we get an answer which is 40 micrometers. What type of microscope was used to obtain the image shown in the diagram above? Give one piece of evidence to support your answer. I hope you can see that it is clearly a 3D image, which means specifically it's a scanning electron microscope. Contrast how an optical microscope and transmission electron microscope work, and contrast the limitations of their use when studying cells. Let's just have a look at the mark scheme here. So this is just a matter of learning these notes off by heart. It's worth 6 marks, so it's worth doing. So as I said, the transmission electron microscope, as we know, is an example of an electron microscope versus the optical one, which uses light. So that's a first comment on how they work. We know that the transition electron microscope allows greater resolution, and that's due to the wavelength of electrons being smaller than that of light. And this means that we have the ability to see smaller organelles with the transition electron microscope, something like a ribosome. And you can get marks by mentioning exact organelles. The transition electron microscope only enables you to see dead specimens. The light one, as I already mentioned, you could see something like the water flea, the Daphnia, and then other comments to do with how thin the specimen needs to be, how complex the transition electron microscope setup is. Something like that is absolutely fine. The cell cycle. So what is the cell cycle? Well, it's a sequence of events which occurs between one cell division and the next. Broadly speaking, it's divided into interphase and mitosis. Mitosis you'll have met before. This is the actual process where cell division occurs, but interphase is also incredibly important, and in fact the cell spends most of its time in the interphase stage of the cell cycle, and there are a variety of steps which make up interphase. But broadly speaking, what takes place in interphase is that the cell is working and operating normally. The organelles are replicated. That's important because obviously any new cells made will need their own organelles. And they're also checked to make sure that replication has occurred correctly. DNA replication occurs in what's known as the S phase of interphase. As I've already said, mitosis is the part of the cell cycle you'll be more familiar with. Mitosis is when the nucleus divides, and there are 4 stages of mitosis. Use PMAT to help you remember the order: prophase, metaphase, anaphase, and telophase. And the last stage of the cell cycle is known as cytokinesis, and this is when the cytoplasm divides, meaning that by the end of it all, you have 2 new daughter cells. So here's some additional content relating to the interphase stage. There are 3 stages of interphase: G1, S, and G2. In the G1 phase, mRNA is made, so that's messenger RNA, as well as proteins needed for DNA replication. Some of the organelles are copied and the cell increases in size. What about the S phase, which comes next? DNA replication takes place in the S phase, and this is the most active part of the cell cycle. G2, there's additional growth of the cell and organelle replication. The DNA replication is checked for any errors, and the cytoskeleton of the cell is dismantled. Throughout interphase, please note that metabolic reactions continue to occur, such as the respiration reactions needed to produce the ATP so that the cell can carry out all of these functions. As I've already said, the cell is constantly checking for errors. These are known as checkpoints, and they occur at each stage of interphase. So the G1 checkpoint checks the cell for its size, any growth factors, nutrients and DNA damage, and if any of these things aren't fine, then the cell enters what's known as G0. The G2 checkput is when the cell size is checked, any DNA replication damage is checked, and any cell damage is also checked. During mitosis, there's actually a spindle assembly checkpoint, and this is to make sure that the chromosomes have attached correctly at the spindle, which is located in the equator of the cell. And this therefore occurs in the metaphase stage of mitosis. We now need to turn our attention entirely to mitosis. So as I've already said, overall what happens in mitosis is that the nuclei divide. That will enable us to produce 2 new cells. Just be aware of the structure of a chromosome. Remember, chromosomes contain long strands of DNA. Each chromosome is made up of 2 chromatids, which are joined at the centre at a structure known as the centromere. So to reiterate, the 4 stages of mitosis in order are prophase, metaphase, anaphase, and finally telophase. So what occurs in the prophase stage of mitosis? So in prophase, the chromosomes condense. This means that they shorten and thicken, and actually they'll be visible under a light microscope. The nuclear envelope begins to break down, and the chromosomes consist of 2 chromatids which are joined at the centromere. And the centrioles become important now because spindle microtubules extend from each of the centrioles, which are located at the ends of the cell, so that they start to reach what's known as the equator of the cell, the middle portion. Metaphase now, so the sister chromatids, which the chromosomes are comprised of, attach to the spindle microtubules via their centromere, and they line up at the equator of the cell. And if you've seen any micrographs, you'll recognize this very clearly. You'll see all those chromosomes lined up at the centre of the cell, its equator. In anaphase, the centromeres divide and the spindle microtubules retract, and that means that the chromosomes start to migrate to opposite poles of the cell. And then last up, telophase. The chromosomes reach the opposite poles of the cell, the spindle microtubule starts to break down, and the nuclear envelopes reform. As I've already said, the final stage of the cell cycle is known as cytokinesis, and this is when the division of cytoplasm occurs. But how does the process of cytokinesis actually take place? Well, microtubules form a ring around the centre of the cell. The microtubules contract, forming what's known as a cleavage furrow, and then the cell is eventually pinched into 2, forming 2 identical daughter cells. Remember, that's characteristic of mitosis. It's a type of cell division which produces 2 genetically identical daughter cells. So that's the process of cytokinesis in an animal cell. But what about the process of cytokinesis in a plant cell? Well, we have slightly different processes here. Excuse me. So here, new cell walls are produced across the equator of the cell using vesicles which contain carbohydrates. The vesicles fuse together to form a cell plate which fuses with the existing cell wall. There's a plasma membrane either side. That makes sense. We know that plant cells contain cell walls and cell membranes. And then finally, the cell divides into 2, forming 2 genetically identical daughter cells. One thing to mention is do get familiar with how the chromosomes appear under a light microscope during the various phases of mitosis. So interphase, you can see here, you can't see the distinctive chromosomes. In prophase, they've started condensing, they've shortened and thickened. In metaphase, as we said, they attach to the spindle microtubules and line up at the equator of the cell. Then in anaphase, the spindle microtubules start retracting, helping to draw those chromosomes to opposite poles of the cell. And then lastly, telophase. We'd expect the next step of cytokinesis to occur. So that's when the membrane breaks down and we get 2 daughter cells. So they often like to use a graph like this in your exam questions. So I'm just going to show you what we can see. So obviously this green line represents the total cell mass. So it makes sense that during the interphase stage and the mitosis stage of the cell cycle that the total cell mass is increasing due to the replication of those organelles and the DNA replication. And then obviously when cytokinesis occurs, which is in effect cell division, you're going to see a rapid decrease in that total cell mass simply because the cell's dividing into 2. If we look at the bottom blue line, which is to do with the mass of DNA, the DNA mass only starts increasing when that DNA replication stage of interphase occurs. So once that DNA has finished replicating, you can see its mass has doubled according to the y-axis scale. And then at cytokinesis, again, when you split your cells, you're obviously going to go down to your original amount of DNA. And the cycle continues. We've mentioned mitosis a lot, but what is its purpose? Well, it's needed for the growth and repair and replacement of tissues, as well as asexual reproduction, which is a specific type of reproduction that only requires one parent's The offspring are clones, they're genetically identical to the parents, and it's a good type of reproduction to use where conditions are unchanging and highly favourable. The mitotic index is the ratio of cells undergoing mitosis compared with the total number of cells in a sample, and it can be calculated by doing the number of cells visible with chromosomes divided by the total number of cells in the sample. Remember, we said that in the first stage of mitosis, prophase, the chromosomes condense and thicken, and that makes them visible. Now, a really nasty disease is cancer, and this is caused by uncontrollable cell growth division. Characteristic of cancer are both benign and malignant tumours. These are huge balls of cells. How do these tumours form? Well, genes controlling mitosis and the cell cycle mutate, causing uncontrollable cell division and growth. A group of abnormal cells known as tumour cells develop and continue to grow. How is cancer usually treated? Well, it makes sense that any drugs we use are used to disrupt the cell cycle. So these drugs are known as chemotherapy, and they actually prevent DNA replication. And if you do A-level chem, you'll have heard of something called cisplatin, which binds to the guanine of DNA, disrupting the DNA structure, meaning that cell division can't occur. Chemotherapy drugs may also interrupt the metaphase stage of mitosis. By doing so, they disrupt the formation of the spindle microtubules, meaning that the chromosomes can no longer attach at the equator, they can no longer migrate to opposite ends of the cell. So that really does disrupt the mitosis stage of the cell cycle. Chemotherapy is great in that it attacks cells which are dividing rapidly. However, it's not the most specific of treatment options. So anywhere else in your body where cells are dividing rapidly are also affected by chemotherapy drugs. And that's why you often notice that people going through cancer treatment have no hair, because chemotherapy affects those cells responsible for producing hair. One thing to notice is that prokaryotes, so these, remember, are cells which do not contain membrane-bound organelles. Good examples include the bacteria. They do not carry out cell division by mitosis because, quite simply, they have no nucleus. Instead, they use binary fission to reproduce. So how does binary fission work in prokaryotes? Well, the circular DNA found in the nucleoid, as well as the plasmids, is replicated, and then the DNA attaches to the membrane. A cell membrane grows between the DNA copies. The cytoplasm then divides, and two daughter cells are produced, each containing the nucleoid, the circular strand of DNA, as well as a variable number of plasmids. What is the equation for the theoretical maximum number of bacteria at a given time? Well, it's given by NT equals N0 times 2 to the N. So NT is the number of cells at any time. N0 is the number of cells when time equals 0. And 2N is the number of divisions. Do note that you can use this equation for calculating the maximum number of dividing cells, for example, in tumour cells. A bacterial cell divides by binary fission every 8 hours. Calculate the number of bacterial cells in a colony formed from one bacterium after 1 week. Assume no bacteria die. So we'll use the equation I just mentioned, where NT equals the number of cells at time t, and 0 equals number of cells at t equals 0, 2n equals the number of divisions. So after NT, what's the number of cells at t equals 0? By definition, that's going to be 1. And then the number of divisions, 1 week, let's get that into hours, 24 times 7. So we know we have 168 hours. We know that each bacterial cell divides by binary fission every 8 hours. So that means we have 21 divisions that took place. So our final answer is 2.10 times 10 to the 6. Now, last up, how do virus particles replicate? Remember, viruses are non-living. They do not contain the cell machinery that we would find in eukaryotes and even prokaryotes. We've already discussed their structure earlier on in the specification. So how a virus replicates is it starts by injecting either its RNA or DNA into a host cell. The host cell replicates the viral genetic material. Viral mRNA is translated into viral proteins, and finally the new virus particles assemble. Describe binary fission in bacteria. So this is when there's replication of both circular DNA, so the nucleoid, and plasmids, and division of the cytoplasm to produce daughter cells. The cell growth rate of the bacterium Bacillus subtilis is proportional to its mass immediately after binary fission. The graph below shows this relationship. The mass of the bacterial cells was measured in femtograms. 1 femtogram is equal to 1 times 10 to the minus 15 grams. Place a tick in the box next to the number that is equal to 680 femtograms. So we'll do 680 multiplied by 1 times 10 to the minus 15. In standard form, that's 6.80 times 10 to the minus 13. So that's there. A scientist determined the growth rate of B subtilis cell by measuring its mass for 5 minutes. In those 5 minutes, the cell's mass increased by 90 femtograms. Use this information in the graph above to determine the mass of this cell immediately after binary fission. Show your working. So let's work out that growth rate per second. So it's 90 femtograms in 5 minutes. What is that per minute? That's 18 femtogram per minute. And then per second, just divide that by 60. So that's 0.3 femtograms per second. And then I'm just going to read across at 0.3, read down. Just read that really carefully, and you can see that's 660. This question is about mitosis in cells. The image below shows the arrangement of the genetic material in a cell during prophase. So that's the first stage of mitosis. Describe and explain the arrangement of the genetic material shown in the above image. So remember, we said in prophase, and actually we can see from that image that the chromosomes have shortened and thickened. So they've condensed, and as a result have become visible. The diploid number of chromosomes in the body cell of an insect species is 4, so that means that the haploid number would be 2, because haploid just means that it contains 1 set of chromosomes. Diploid means it contains 2 sets. Tick the box next to the diagram that represents the appearance of chromosomes in a cell during metaphase in this species. So they're going to line up at the equator. They're going to be attached by the centromere. So the answer here is A. The figure below shows the mean distance between centromeres and the poles of the spindle during mitosis. Calculate the rate of movement of the centromeres during phase E. So zooming in on this phase, my friend's kitten meowing. Hi, Tammy. Tammy. So the rate of movement is really the distance moved over time. That's what rate implies, i.e. the speed. So we'll do the distance, which was 19, down to here it's 4, over the time frame. So the time frame is 700 seconds. So that's the rate of movement in micrometers per second. I'm using these units to help me here. Just be careful, it says it wants it per minute. And then multiply that number by 60 to get it per minute. So 1.286 is our final answer to 3 decimal places. Name the 3 phases of mitosis shown by C, D and E on the figure above. Describe the role of the spindle fibres and the behaviour of the chromosomes during each of these phases. So make sure you double check what the figure is showing you. It's the mean distance between the centromeres and the poles, the ends of the spindle during mitosis. Remember that this is your centromere, these are your 2 poles, and this is your equator. So remember, if these are your 2 poles, remember that in metaphase, the chromosomes line up at the equator, and then in anaphase, they start migrating towards the poles. So that distance will therefore reduce. So we can say that this is anaphase, this is metaphase, because of the equator, and this is prophase. So you get marks for saying exactly what each of these phases are. And then make sure you're answering the question in terms of what's happening to the spindle fibres and the behaviour of the chromosomes. So we've kind of dealt with that graph. We just need to list exactly what happens in each of the stages. So in prophase, as we've said, the chromosomes condense and become more visible. In metaphase, chromosomes line up at the equator of the cell. In anaphase, the centromeres divide and sister chromatids migrate to opposite poles of the cell. And then, because it mentions the spindle fibres, we're going to say due to the spindle microtubules retracting. So that's a nice full answer in order to get those 5 marks. Figure 1 shows the cells the student saw in one field of view. He used this field of view to calculate the length of time these onion cells spent in anaphase of mitosis. Scientists have found the mean length of time spent by onion cells in anaphase of mitosis is 105 minutes. They also found the cell cycle of cells in the onion root shown takes 1080 minutes. 32 whole cells are shown in Figure 1. Use this information in Figure 1 to calculate the length of time the cells of this onion root are in anaphase, and then calculate the percentage difference between your answer and the mean length of time found by the scientist. Show your working. Okay, do take your time to work through all this info. So our first step is to look at these 32 cells and work out how many of the cells are in anaphase. Remember, that's when the chromosomes start migrating to opposite poles of the cell. So that's that cell, and this cell, so 3 cells. So if that's 3 cells out of 32, as a percent we do 3 divided by 32 times by 100. So we know that 9.375% of the cells present here are in anaphase. So how much time does that take? We'll find 9.375% of 1080. So my calculations state that it's going to take 101.25 minutes, but the scientists thought that it would take 105, so just work out the difference between those numbers, which is 3.75. And then annoyingly, because it's the percentage difference, we've got to do an extra step, which is when you do the change, so that's 3.75 over the original time, which was 105, multiply it by 100 to get a percentage difference of 3.57%. Take the name given to the division of cytoplasm during the cell cycle, that is cytokinesis. Sorry, that's the kitten. But it's her home, so she can do what she likes. Figure 1 shows all the chromosomes present in one human cell during mitosis. A scientist stained and photographed the chromosomes. In Figure 2, the scientist has arranged the images of these chromosomes in homologous pairs. Give 2 pieces of evidence from Figure 1 that this cell was undergoing mitosis. Explain your answers. Your first point is to say that the individual chromosomes are visible because they have condensed. And the other thing you want to point out is that each chromosome is made up of 2 chromatids, and that must be because the DNA has replicated. Tick one box that gives the name of the stage of mitosis shown in Figure 1. Because it's simply the condensation stage, that's going to be the first stage, so prophase. When preparing the cells for observation, the scientists placed them in a solution that had a slightly higher, less negative water potential than the cytoplasm. So the surrounding solution, in effect, had a higher water concentration. This did not cause the cells to burst, but moved the chromosomes further apart in order to reduce the overlapping of chromosomes when observed with an optical or light microscope. Suggest how this procedure moved the chromosomes apart. So you are bathing your cells. Here's your cell. Here's your surrounding solution, which we know contains more water. It has a higher water potential. So water enters those cells by osmosis from an area of high water potential to an area of low water potential. By doing that, it means that that cell gets bigger, meaning that the chromosomes move apart. The figure below shows some cells from an onion root tip at different stages of the cell cycle. Place stages A to E in the correct order. Start with stage D. Then we need our chromosomes to condense, which is C. Then they need to line up at the equator, which is B. Then they need to migrate to opposite poles, E. And then cytokinesis takes place, so that's A. Today we're moving on to cell membranes and transport, so we'll be looking in great detail at the fluid mosaic model. But first of all, we need to ask ourselves, why do cells require membranes in the first place? Well, cell membranes provide a partially permeable barrier between cells and their environment. They also provide a partially permeable barrier between organelles and the cytoplasm, and they enable the isolation of the contents of vesicles. And that's important if you think that many vesicles contain hydrolytic enzymes that are incredibly destructive. They need to be used in a very particular way in order to destroy pathogens. So it's absolutely essential that these hydrolytic enzymes are enclosed safely in vesicles made out of membranes. Cell membranes also provide an attachment surface for enzymes. They are sites of cell communication, and they create concentration gradients within organelles. Now, over time, we've had many different models of cell membranes. One of the things which is common to many cell membrane models is the existence of a phospholipid bilayer. However, it's actually the placement of proteins which varies greatly. And the current model which is accepted is known as the fluid mosaic model. So where does that name come from? Well, the fact that the word mosaic is present means that the cell membrane consists of many different components, such as phospholipids, carrier proteins, glycoproteins, cholesterol. And the fluid aspect means that these components are free to move within the phospholipid bilayer. Now, phospholipids are known as amphipathic, and that means that one end of them is attracted to water, so that's hydrophilic, the other end is repelled by water, so is hydrophobic. So why do phospholipids exhibit amphipathic behaviours? And that's very much due to their structure, which comprises of a phosphate head and two fatty acids tails. Now, the phosphate head is polar, and this makes it hydrophilic, water-loving. It means that it faces outwards towards the cytoplasm or tissue fluid. The fatty acid tails are non-polar, therefore they're hydrophobic, so they face inwards. So what is the role of phospholipids? Well, they allow lipid-soluble substances to enter and leave the cell. They prevent water-soluble substances from entering and leaving the cell. And they make the membrane fluid, which means that it's self-healing and flexible. So I've already mentioned that there are cholesterol molecules present, but what is the role of the cholesterol? Well, it affects the fluidity of the membrane. It helps restrict the movement of the phospholipid molecules and stops the membrane becoming too fluid when it heats up, as well as preventing it becoming too stiff when it gets cold. Cholesterol molecules also reduce the permeability of the membrane. Now, there are many different types of protein present in the membrane. Some of these proteins act as receptors for neurotransmitters and hormones. Some of the proteins act as carrier molecules. These are known as channel proteins, which are used in both facilitated diffusion and active transport. Now, channel proteins are used for facilitated diffusion, whilst carrier proteins are used for active transport. Other proteins are used in cell adhesion. Some act as enzymes. Glycolipids now, these have both a carbohydrate and lipid element. These act as recognition sites, and they help maintain the stability of the membrane. They also assist with adhesion, which helps cells stick together in order to form tissues. What is the role of glycoproteins? Well, in a similar way to glycolipids, they can act as recognition sites, helping lymphocytes, which are a type of white blood cell, recognize its own cells. Glycoproteins act as receptors for cell-to-cell communication. This is known as cell signaling. Again, they can help with adhesion, so helping cells stick together to form tissues. And glycoproteins can also act as antigens. Now, antigens, remember, are small proteins which are recognized by white blood cells. You'll have come across antigens on pathogens before, such as on bacteria, and it's these antigens which lymphocytes recognize so that they can produce antibodies. Let's have a look really closely at this fluid mosaic model. So as I've already mentioned, here's our phospholipid with its hydrophilic phosphate head. That's due to the phosphate being polar, so therefore the hydrophilic head is attracted to water. Here are your two fatty acid tails, which are non-polar, so therefore water-hating, hydrophobic. If you look at the more detailed structure of the membrane, here's your phospholipid bilayer. The phosphate heads point outwards, the fatty acid tails point towards each other. You've got a large number of different proteins. You have an intrinsic protein here, which pokes into the cell. You've got your protein carrier. Here's an extrinsic protein. The cholesterol tends to be shown as just a small set of molecules. Here, remember that is going to affect the fluidity of the membrane. Here we have glycolipids. This is a glycoprotein which has the carbohydrate portion as well as the protein portion. What about the effects of temperature on the membrane structure? So temperature is largely going to affect the phospholipid bilayer as well as any proteins which are present. That makes sense really, because remember, proteins can be denatured by excessively high temperatures. Now when you increase the temperature, the phospholipids start to move more, and that increases the fluidity of the membrane, creating gaps. As a result, this increases the permeability of the cell membrane, and if that temperature is raised too high, then the cell membrane can be damaged irreparably. Now let's look at the effects of various solvents on the cell membrane structure, starting with water, which is an example of a polar solvent, because remember it has that electron deficient hydrogen and the electron rich oxygens. Now water is responsible for the orientation of the phospholipids, because remember their polar phosphate heads are hydrophilic, their non-polar fatty acid tails are hydrophobic. So water is responsible for the phospholipids arranging themselves into a bilayer, which just means a double layer. As you'd expect, the hydrophilic heads face outwards, the hydrophobic tails face inwards towards each other. So water as a solvent does not disrupt cells. Now organic solvents, which are less polar than water, dissolve membranes and therefore cause massive damage to cells. Now we're being asked to describe an investigation into the effect of temperature on membrane permeability. So a good way of doing this is by using beetroot, where we're going to obtain equal sizes. We're also going to wash it in order to remove all the surface pigment released from damaged cells. We then place pieces in boiling tubes containing 100 ml of distilled water, place the boiling tubes in a water bath, and then increase the temperature of the water bath in 10-degree intervals. Take a sample of water 5 minutes after each temperature increase. This allows the mixture to equilibrate. And then finally measure the absorbance using colorimeter with a blue filter. The colour filter you need to use will vary depending on the colour of the substance, but blue is appropriate for the red beetroot, as I've said here. And then as the temperature increases, the membrane permeability increases and more pigment is released from the cells due to damage to that membrane, so the absorbance measured will increase. Then we're going to repeat 3 times to improve the reliability. So here our independent variable was the temperature, that's what we were changing. What we were measuring is the amount of absorbance as measured by the colorimeter. How could this procedure be adapted to investigate the effects of different solvents on membrane permeability? Well, this time we need to keep our temperature constant, but we're going to use a range of different organic solvents as our independent variable. And then lastly, what is the effect of an acidic solution on cell membrane? It denatures the cell membrane proteins because it has a very low pH, and that's therefore going to increase the permeability of the membrane. So what are the different ways in which particles can move across membranes? Well, you've met simple diffusion, osmosis, and active transport. You may be less familiar with facilitated diffusion as well as bulk transport, which is either exocytosis or endocytosis. The definition of diffusion you'll know off by heart. It's the net movement of particles from an area of high concentration to an area of low concentration. This occurs down the concentration gradient. It's a passive process, so there's no input of energy from ATP. And diffusion continues until the concentrations are equal on each side. Facilitated diffusion still has diffusion in its name, so again it's a passive process, no need for ATP, and particles still move from an area of high concentration to an area of low concentration. However, the facilitated aspect means that it needs a little bit of help from a carrier protein which allows only specific substances through. Glucose is a good example of a substance which moves by facilitated diffusion, whereas gases like oxygen and carbon dioxide move by simple diffusion. As we move on to osmosis, it's important you know the following definitions. So what is solute? It's a solid which dissolves in a solvent. What is a solvent? The liquid in which the solute dissolves. Solution is a mixture of a solute and a solvent. At GCSE, you might have met the term water concentration. You might even have met the term water potential. That's good news, because in high-level bio, we need to use the term water potential, which describes the pressure exerted by water against a container or a membrane. And this is due to the water molecules colliding with said membrane or container. It's a pressure, so it's measured in kPa. By definition, The water potential of pure water at standard temperature and pressure is 0 kPa. So what is the definition of osmosis? Well, it's the net movement of water molecules from an area of high water potential, so that means an area with high water concentration, to an area of low water potential, low water concentration, across a partially permeable membrane. So what happens if we add salt to our solution, so we increase the amount of solute? Well, that creates a more negative water potential. If we place animal tissue in a concentrated solution, so one with a more negative water potential, one that contains lots of salt, there'll be a net movement of water from inside the animal cell to the surrounding solution by osmosis across a partially permeable membrane. The animal cell becomes crenated, which in effect means shriveled or wrinkled. If you place a plant cell in a concentrated solution, so one with a low water potential, again water leaves the plant cell by osmosis across a partially permeable membrane. The appearance of that plant cell will be plasmolysed, and that means that the cell membrane pulls away from the cell wall and overall it appears flaccid. What about if we switch around and we have a low solute concentration, so something like pure water, which has a very high water potential? When an animal cell is placed in a high water potential solution, water is going to enter the animal cell by osmosis across the partially permeable membrane. Because the animal cell has no cell wall, if enough water enters, the animal cell will burst. For a plant cell, it's not so dramatic. Again, water enters the plant cell by osmosis, but due to the presence of that plant cell wall, you'll find that the cell becomes swollen. This is known as turgid. If you're asked to describe a method to investigate osmosis in living tissue, particularly plant cells, you might have done this before using potato chips. Use a core borer to obtain potato chips of the same mass and the same surface area. You're going to place these potato chips in different concentrations of salt solution, and then you're going to measure the mass beforehand. Then you place your potato chips in the various salt solutions for 24 hours. Then you remove the potato chips, dry them with a piece of kitchen towel in order to remove excess solution, and then weigh them. It's important that you calculate the percentage change in mass rather than the change in mass, and that enables valid comparisons to be made in the case that you weren't able to make the starting masses exactly the same. Remember, you then plot your results on your graph, so on your y-axis, because remember that is your dependent variable, what you're measuring, that will be your percentage change in mass. Your x-axis, that's what you're changing, your independent variable in this case will be the concentration of salt solution. By plotting your graph, you'll be able to determine the exact concentration present in the potato at the point where the graph line crosses the x-axis, because you'll have a 0% change in mass. Moving on to active transport. So this is the net movement of particles from an area of low concentration to high concentration. Because this occurs against the concentration gradient, we say that it is an active process and therefore requires energy from ATP. Often it also requires the presence of protein pumps. Now, these protein pumps are highly specific, working on one substance only, and they work in one direction only. The specific substance enters the protein pump from the low concentration side. Energy from ATP changes the shape of the pump, and then the pump releases your substance to the side with higher concentration, and finally the pump returns to its original shape. Co-transport involves the movement of two molecules at the same time. They use the same transport protein. A good example here is the movement of sodium ions and glucose in the ileum of the digestive system. This often requires the use of energy which is released by the hydrolysis of ATP. So the two types of bulk transport, which basically means moving larger substances, are known as endocytosis and exocytosis, and which one of these occurs totally depends on which direction the substances is moving. So endocytosis is all about moving substances into the cell. The plasma membrane encloses the solid particles, and then it forms indentations, and finally the membrane pinches off, and this does involve ATP. A vesicle forms, and so it makes sense that the cell membrane has become the outside of the vesicle. And finally the vesicle breaks away from the plasma membrane, releasing the contents into the cell. So what is exocytosis? Well, the opposite is going to take place here. So in this case, the vesicle moves towards the plasma membrane. This requires ATP. The vesicle fuses with the plasma membrane and releases its contents to the outside of the cell, and the membrane returns to its original shape. And importantly, it's the fluidity of the membrane which enables both exocytosis and endocytosis to take place. But how are these vesicles moved about the cell? Well, it involves the use of microtubules which form a cytoskeleton which the vesicles can move along. These microtubules can be extended as well as being broken down, but it does require ATP in order to do this. Let's now mention exact examples of various substances which are going to move into and out of the cell and the method by which they do this. So let's start with small non-polar substances like carbon dioxide. These will simply diffuse across the membrane by simple diffusion. Larger substances use bulk transport such as endocytosis. This could be a pathogen being enclosed inside a vesicle, which then enters a white blood cell such as a phagocyte where it can be destroyed. Polar substances may move by facilitated diffusion, for example glucose, which uses a channel protein. Other polar substances move by active transport, which require carrier protein. And an example of this is the sodium-potassium ion pump. And last up, and probably most straightforward, is the movement of water, which obviously occurs by osmosis. So how can the transport rate of these substances be increased? Well, we can increase the surface area, such as the presence of villi and microvilli in the ileum of the small intestine. By increasing the surface area, it means you've got more of an area for diffusion to occur across, as well as more space for the presence of transport proteins needed in active transport and facilitated diffusion. Linked with this, we can increase the transport rate by having more transport proteins, and we can also increase the steepness of the concentration gradient. That will especially affect how quickly simple diffusion, facilitated diffusion, and osmosis occur. In 1935, scientists suggested a model for the chemical structure of a cell surface membrane. Figure 2 shows the membrane structure the scientists suggested. So this is our old model. It's not going to be entirely correct. Give one similarity and two differences between the membrane structure shown in Figure 2 and the fluid mosaic model of membrane structure. So what can we see that's the same here and what's different? So actually this phospholipid bilayer is obviously very, very similar. There's one layer, there's the second layer, hence bilayer. You've got your phosphate heads and your fatty acid tails. There's two of them. Remember, these are the hydrophobic ends, these are the hydrophilic ends. So in that way, they're very similar. I'm just going to say both have a phospholipid bilayer. You could have mentioned those things to do with the phosphates or the fatty acids. In terms of differences, I think obviously key here is the location of these proteins. The fluid mosaic model, the modern day model, has carrier proteins, and the proteins are dotted within the membrane, whereas here you can see a single protein layer on the outside. What is absent about this model is it doesn't include any cholesterol or glycoproteins or glycolipids. So up to you again in terms of what differences. So I'm going to say the old model contains no cholesterol, the old model contains no glycoprotein. The diagram below shows part of a plasma membrane. Which of the label lines points to a structure that could contain a sulfur atom? So sulfur is sometimes found in protein. So we need to look at this closely to see which of these is a protein. To me, this looks like a glycoprotein, meaning that there's a carbohydrate portion and a protein portion, so that would be one only. The diagram below shows the structure of a plasma membrane. Which label, A to D, indicates the component of the membrane that can affect its fluidity? Remember, that's cholesterol. So that's D. The movement of substances across cell membranes is affected by membrane structure. Describe how. So we're going to talk about the different components of the cell membrane and then describe which substances can move across. So the phospholipid bilayer allows the diffusion of, what did we say, carbon dioxide and oxygen, but prevents the movement of polar substances. We're going to say that channel proteins allow the facilitated diffusion of, what substance did I mention? It was glucose. So you do need to be quite in depth here with your knowledge. You can even point out that cholesterol affects the fluidity of the membrane and therefore the processes of exocytosis and endocytosis. Based on the wording of the question where it says the movement of substances across cell membranes is affected by membrane structure, we can also talk about the factors which influence this overall, such as the surface area. So membrane surface area determines how much diffusion can take place. Sodium ions from salt, sodium chloride, are absorbed by cells lining the gut. Some of these cell membranes have a carrier protein called NHE3. NHE3 actively transports one sodium ion into the cell in exchange for one hydrogen ion out of the cell. Use your knowledge of transport across cell membranes to suggest how NHE3 does this. So because we've got one sodium ion moving in, one proton moving out, this is an example of co-transport. And therefore requires the use of ATP. So what happens is the sodium ion and hydrogen ion bind to a protein and the protein changes shape to move the H+ ion and sodium ion across the membrane. Let's crack on with the immune system. So we're going to start with our definition of a pathogen. This is, simply put, a microorganism which causes disease, such as a virus or bacteria. Now, on the outside of them, they often have what's known as an antigen, and this is recognised as our bodies as being non-self. These antigens are often made out of proteins, and it's because there's a huge variety of proteins due to their complex 3D structures held together by those bonds in the tertiary structure that accounts for this huge variety. So what else do we consider as being non-self? So as I've already said, pathogens, also toxins, cancer cells, and cells from other organisms of the same species. We have several different defense systems that work to attack pathogens. The first one is the non-specific defense system. Its aim is to break down or prevent the entry of pathogens. And examples of non-specific defense mechanisms include our skin acting as a barrier, mucus in our trachea trapping pathogens and then being removed through the action of the ciliated epithelial cells. You've got the production of enzymes and hydrochloric acid, which remember is produced by your stomach. Phagocytes' role is to engulf or ingest pathogens. Blood clotting at wounds occurs, as well as many different inflammatory responses. So I've already mentioned them lots, but what sort of things do you need to know about phagocytes? Well, as I've said, they destroy pathogens by ingesting them. They do this primarily by squeezing out of capillaries so they can reach the pathogen. Then they enclose the pathogen in a process known as endocytosis, which remember is a type of bulk transport. The phagocytes then secrete hydrolytic enzymes which destroy the pathogens, and remember this is an example of a non-specific immune response. So in greater detail, first of all, the phagocyte needs to be attracted in a process known as chemotaxis. The phagocytes have specific receptors on their membranes which are complementary to the antigens on pathogens. In a process known as phagocytosis, the foreign matter is engulfed by the phagocyte and it gets enclosed in what's known as a phagosome. The phagosome fuses with vesicles known as lysosomes to form a phagolysosome. The lysosome contains lysosome enzymes, hydrolytic enzymes, which destroy the pathogen. And the products of this destruction are absorbed into the cytoplasm. Now, one thing to be aware of is that at this stage in high-level bio, we're going to refer to phagocytes as macrophages, so they are part and parcel of being the same thing. So at this point, we know that the phagocyte has engulfed our pathogen. It's broken it up into small pieces, which are now located within its cytoplasm. Now, what happens next is special molecules known as MHCs major histocompatibility complexes are important. And these are glycoproteins found in the cytoplasm of these macrophages. They combine with the antigens from our pathogen and they cause the phagocyte, the macrophage, to express these antigens on its surface. So at this point, these cells are known as APCs, antigen-presenting cells. So to summarise, what is the role of our phagocytes? Well, it's to engulf and digest pathogens. It's to display the pathogen's antigens on its surface, turning it into an APC, an antigen-presenting cell. And lastly, it's to activate T cells. And we'll talk about that very shortly, I promise. Describe how a phagocyte destroys a pathogen present in the blood. So we know that phagocytes engulf pathogens which are then enclosed in a phagosome. Phagosome fuses with lysosome to form phagolysosome. Lysozyme enzymes released and pathogen is digested. Give two types of cell other than pathogens that can stimulate an immune response. So we said cancer cells and we said cells from other organisms of the same species. So we're going to move on to the role of various T cells and B cells. So that's T lymphocytes and B lymphocytes. Where do they get their names from? Well, it's based on where they're made. T cells are made in the thymus, whilst B cells are made in the bone marrow. So the specific immune response is divided into two, the cell-mediated response and the humoral response. The cell-mediated response, as the name suggests, a response controlled by cells, and the type of cells that are important here are known as T cells. We have T helper cells, T killer cells, and T memory cells. So what is the role of the T helper cells? Well, they have receptors on their surface known as CD4 receptors, and these bind to antigens on our antigen-presenting cells. So remember, that's our macrophages, which are displaying the pathogen's antigens. They secrete interleukins, cytokines, which activate B cells, as well as other T cells. So that's why they're known as T helper cells, because they're going to, in essence, activate other useful cells. Now, in terms of the way that the T helper cell responds to a particular APC, there's only certain T cells which are complementary to the antigens displayed by the APC. And obviously only these T helper cells will be appropriate in fighting the pathogen. So once the correct T helper cell has been selected, it divides by mitosis in order to produce more copies, so you end up with many more of these appropriate T helper cells. What about our T killer cells? Really, we need to be calling these cytotoxic T cells. They produce a substance known as perforin. As the name suggests, they perforate the cell membrane of our pathogens. In this way, they help destroy them. They produce small holes in that cell surface membrane. The T memory cells use their name to help you remember what they do. They provide immunity, they circulate in our blood, and they recognize those antigens far more quickly if there's re-entry of a particular pathogen. And the T regulator cells, a fourth type of T cell, control our immune response once the pathogen has been eliminated. So as I've said, the response of the T cells is an example of cell-mediated immunity. It's T cells responding to a change in an organism's own cells. So that could be the introduction of a pathogen, it could be cancer cells, it could be transplanted tissue, as well as the antigen processing phagocytes. Obviously, we know those T cells are responding to the APCs. Moving on to the second type of specific immune response, the humoral response. So starting with our B lymphocytes, our B cells, the ways in which they can respond is, first of all, they can respond directly to a particular pathogen's antigens, or they'll respond to the cytokines and interleukins produced by T helper cells. So some of our B lymphocytes have antibodies on their cell surface membrane, and as we know, antibodies are specific or complementary to one type of antigen. The B lymphocyte is able to engulf and digest the pathogen, and then it's able to present the antigen to again become an APC, an antigen-presenting cell. An activated T helper cell can then bind to that B cell APC, which is known as clonal selection, and then it undergoes mitosis to produce many more copies, known as clonal expansion. A type of B cell known as plasma cells are responsible for secreting antibodies. And then B memory cells are important because they provide, in effect, a memory which enables a much faster response upon re-entry of that pathogen. So how do the cytokines and interleukins produced by the T helper cells help relevant B cells to divide? Well, the cytokines and interleukins have a specific shape which binds to a particular B lymphocyte, and this activates the clonal expansion of the B cells. Collagen is a protein produced by cells in joints such as the knee. Rheumatoid arthritis is an autoimmune disease. In an autoimmune disease, a person's immune system attacks their own cells. Rheumatoid arthritis causes pain, swelling, and stiffness in the joints. Scientists have found a virus that produces a protein very similar to human collagen. Suggest how the immune response to the viral protein can result in the development of rheumatoid arthritis. That viral protein is going to act as an antigen, and then specific B cells undergo mitosis to form both plasma cells, which release antibodies, and B memory cells. Antibodies cause symptoms of rheumatoid arthritis. So we know that antibodies form a large part of this specific immune response in order to deal with pathogens. Let's now discuss the structure of antibodies. So they have a characteristic Y-shaped. So I'm actually going to use my arms to represent that. If you take my whole body with my arms held up as being a Y shape, just like the antibody, remember that this antibody is made out of protein, so it has a very specific 3D structure, and indeed its structure is held in place through the presence of disulfide bridges. But taking that Y shape again, you have two chains known as heavy and light chains. I want you to think of my arms as being the light chains, and that the outer portion of my arms are the two light chains of the antibody. The heavy chains sit on the inside of my arms and run all the way down to my feet, and they're going to form what's known as the heavy chains. After all, they're much longer than the light chains of my arms. Then you have what's known as a variable region located at the top of the Y. This is complementary to specific antigens on pathogens. You have a constant region at the bottom of that Y, so at the bottom of my feet, which is the same for all antibodies, and it helps our bodies recognize the antibodies as being self, so a part of us. The hinge region is located at the bottom part of the Y. It enables the antibody to move in order to bind to the complementary antigen. So how can antibodies help fight infection? Well, first of all, they can produce antitoxins, which help to neutralize the toxins produced by pathogens. They cause agglutination, which really means they help the pathogens clump together, making them more recognizable by phagocytes so they can be engulfed. They also act as opsonins, and again, this makes it easier for the phagocytes to recognize them. So looking at the process of agglutination in more detail, we know that that means when the antibodies cause the pathogens to stick together. This means that they're now too large to enter the host cell, therefore increasing the likelihood of them being engulfed by phagocytes. The diagram below shows the structure of an antibody. Label the diagram above with an X to show where an antigen-antibody complex forms. So this is your light chains, your heavy chains, your hinge region, your constant region, and your variable region. That variable region means that you're going to have a variety of different antibodies which are complementary to antigens, so that is where your X needs to be. A disulfide bridge is labelled in the diagram above. What is the role of the disulfide bridge in forming the quaternary structure of an antibody? Remember, antibodies are made from proteins, so the disulfide bridge joins two polypeptides. Describe and explain the role of antibodies in stimulating phagocytosis. Do not include details about the process of phagocytosis. So how do antibodies increase the likelihood of phagocytosis taking place? Well, remember, they cause agglutination, where they bind together many of the pathogens. So the clump of pathogens is more likely to be engulfed by phagocytes. antibodies act as opsonins, which make it easier for the phagocytes to recognise the pathogens. Meningococcus bacteria cause a disease called meningitis. Scientists investigated a new meningitis vaccine by measuring changes in blood anti-meningitis antibody concentration in mice. Each mouse was given 3 separate Mengi injections. The concentration of anti-meningitis antibodies was measured in a sample of blood taken soon after each injection. After the 3rd injection, the concentration of anti-meningitis antibody in the blood was also measured after 60 days, 120 days, and 180 days. The graph shows the scientists' results. So on the y-axis, we have the concentration of anti-meningitis antibodies. Remember, each point represents a different mouse. And then here we have the concentration of antibody 60, 120, and 180 days after the 3rd injection. Use the key. The protective antibody concentration is given here, whilst the average anti-meningitis antibody concentration is given here. The scientists discovered that the concentration of anti-meningitis antibody of the mouse labelled Z in the graph decreased after the 3rd injection at a constant rate of 0.027 arbitrary units per day. Use this information in the graph to calculate the number of days after the 3rd injection. The antibody concentration is higher than the protective antibody concentration for this mouse. So the first thing I've done is labelled the concentration of the protective antibody, which is 2.1. What about the concentration of Z? From the label of Z, we can see that the concentration of antibody here is 5.1. If you do chem, you'll know that this means concentration, the square brackets. And then here we're told that the concentration decreases by 0.027 units per day. So we need to work out how many times 0.027 goes into the difference between these two values. So there's the difference between those two values divided by 0.027 equals 111 days. Although we've mentioned this in passing, what is the role of memory cells in our immune response? We're going to look at both the T memory cells and B memory cells. In essence, both of them recognise the pathogen upon re-entry. They produce clones through the process of mitosis. Now, the B cell clones produce plasma cells. Remember, plasma cells are responsible for making antibodies, whilst T cell clones can produce T helper cells and T killer cells or T cytotoxic cells. And we say that these memory cells are responsible for the secondary response. Again, that means when that pathogen has re-entered the body. If we look at this graph, which takes antibody concentration versus time, we can see that upon first exposure to the pathogen, antibodies are produced slowly and in a small quantity. When that pathogen enters a second time, antibodies are produced much faster, sooner, and in greater quantity. So what is the principle of immunity? Well, it's the principle whereby our bodies are able to resist infection, and that's due to the presence of antibodies. So the pathogen enters the body. This leads to the clonal selection of B cells. The B cells produce specific antibodies, and if the same pathogen enters the body again, memory cells are activated, which produces antibodies faster, sooner, and in greater quantity. So the various types of immunity we need to look at. Passive immunity means acquiring antibodies from external sources such as breast milk and across the placenta and through the injection of antibodies. Active immunity is when our body produces its own antibodies, so that would be the case of an active infection or through vaccination. Natural immunity, as the name suggests, occurs naturally within the body. Artificial immunity is when you get immunity due to intentional exposure to a particular antigen. And this table will now show you specific examples of each. So you can see that the major advantage of active immunity is that it produces B and T memory cells and therefore leads to long-lasting immunity, whereas passive immunity does not produce memory cells and so therefore you don't have a faster secondary immune response. Obviously, it looks like active immunity is better than passive immunity, so why do we still bother injecting antibodies? Because that will still work faster than waiting for an immune response. Describe the difference between active and passive immunity. We need 5 separate points here. Let's use this space down here to make a quick table comparing them. See if we can find 5 separate points. Obviously, the first thing to be aware of is that passive immunity involves receiving antibodies from external sources, such as across the placenta from breast milk, the injection of antibodies, such as the case of antivenom, whereas active immunity is when our body produces our own antibodies from facing an infection directly or through vaccines. So first of all, let's really say that. What about how long these types of immunity last? Well, active provides long-term immunity, passive provides short-term. Do each of these involve memory cells? With active immunity, the answer is yes, due to the B cells undergoing cell division by mitosis to form memory cells. In passive immunity, that won't take place. How quickly do these types of immunity occur? Well, active is slow acting because you have to wait for those B cells to divide, whereas with passive immunity, the antibodies are already there, such as the case of antivenom, so it's much faster acting. For your fifth mark, you would say that with active immunity, that the antibodies are made by plasma cells. Now I want to explain how vaccinations work. So remember, vaccines are examples of artificial active immunity. So in terms of what happens when you receive a vaccination, you receive an injection containing a weak, attenuated or dead form of your pathogen. That's important because after all, you don't want to have an active infection, one where you feel physically ill. Now, antigens will activate the specific immune response. So that means the response involving B cells and T cells. The T cells are activated through clonal selection because remember, it's only very specific T cells which will be complementary to the antigens on the pathogen. And those T cells, the T helper cells, stimulate the clonal expansion of B cells. Memory cells are made and specific antibodies are produced by B cells. The result is a specific immunity. And so overall, vaccination results in the slow production and a lower level of antibodies. And then upon re-entry, so if you get exposed to that pathogen again, what happens is you're going to produce antibodies much quicker, sooner, and in greater quantity. You'll often find that you receive booster shots of your particular vaccination, and that's in order to stimulate memory cells. 2 words to do with epidemiology now. So we're looking at the words epidemic and pandemic. So an epidemic is when a communicable disease, so one that passes from person to person, spreads rapidly at a local or national level. Whereas a pandemic is when a particular communicable disease spreads rapidly on a global scale. And obviously the disease on our mind is COVID, the coronavirus. So how do vaccination programs help prevent epidemics? That's because mass vaccination can prevent the spread of a disease on a wider scale. And what it means is that something known as herd immunity is brought about. And that's when a significant number have been vaccinated. It gives protection to those who have not got immunity. Now, vaccinations have to be changed regularly. You might be aware that the flu vaccine is a different vaccine every single year. And the reason for that is because pathogens, antigens mutate regularly. This produces different strains with different antigens, and therefore new vaccination is needed for those new strains. So which type of people are considered particularly vulnerable when it comes to creating a vaccination program? These are the people we'll target first. That will be the elderly, the immune compromised, so maybe people that have received an organ transplant, pregnant females, those with chronic diseases such as kidney disease, as well as health workers. So what are the features of a successful vaccination program? Cost is important. It needs to be economically viable for most of the population to receive a vaccine in order to produce that herd immunity that I just mentioned. The vaccine should have few side effects after all. If it produces unpleasant side effects, people won't want to receive it. The production, transport and storage of the vaccine is essential. If it needs to be kept refrigerated, that's certainly something to take into account, particularly in places around the world that are hot and have poor infrastructure. You need the presence of trained healthcare workers who can administer the vaccination. So why might a vaccination program prove unsuccessful? Well, primarily if it fails to produce immunity in some individuals. If the individuals become infected with the disease after the vaccination, that's a huge failure. They're going to feel very unwell indeed. And that can be if their immunity levels are so weak that they are still susceptible to the disease even in its weakened form. Another reason why a vaccination program may not eliminate a disease is if you're working with a particular pathogen whose antigens mutate regularly, such as influenza. And that means that the antigens are no longer recognized by the immune system. Another reason could be because the pathogen itself hides inside our own cells, such as HIV, which, remember, infects our lymphocytes. So there's obviously no way of our immune system reaching that, and ethical reasons may also reduce take-ups. Describe how the B lymphocytes of a frog would respond to vaccination against ranavirus. You can assume that B cells of a frog respond in the same way as B cells of a human. Do not include details of the cellular response in your answer. So start by talking about the fact that the B cells respond to the antigens present in the vaccination. Complementary B cells undergo clonal selection and divide by mitosis, and then we need to say B cells develop into memory cells, whilst other B cells, plasma cells, secrete antibodies against the virus. When a person is bitten by a venomous snake, the snake injects a toxin into the person. Antivenom is injected as treatment. Antivenom contains antibodies against the snake toxin. This treatment is an example of passive immunity. Explain how the treatment with antivenom works and why it is essential to use passive immunity rather than active immunity. This point here is obviously all to do with timing. The passive immunity is about injecting antibodies, which will actually attack the toxin and cause its destruction, whereas active immunity would take far too long waiting for our B cells and T cells to get involved, and at that point that person might have unfortunately already died, depending on how venomous that snake is. So let's start by saying that antivenom antibodies bind to the toxin and cause its destruction. Active immunity would be too slow and may lead to the death of the person. During vaccination, each animal is initially injected with a small volume of venom. 2 weeks later, it is injected with a larger volume of venom. Use your knowledge of the humoral immune system to explain this vaccination program. So the humoral immune response is to do with B cells. So the first thing we want to say is that B cells specific to the venom undergo clonal selection and divide by mitosis, clonal expansion. Then the next step is for B cells to produce both memory cells and plasma cells. Those plasma cells are responsible for making antibodies. What's the purpose of the larger volume of venom being injected 2 weeks later? Well, that means that as a result of that second dosage, those antibodies, which form part of the secondary immune response, will be produced far quicker, far sooner, and in much greater concentration. And it also makes sense that you want to keep that first dose nice and low because, after all, you don't want to accidentally kill the animal in question. Now we move on to AIDS, which stands for Acquired Immunodeficiency Syndrome. Now, AIDS is the disease, but how is it caused? It's caused by a particular virus known as HIV, which stands for Human Immunodeficiency Virus. HIV is an example of what's known as a retrovirus. That's because it contains RNA and a reverse transcriptase enzyme, hence the name retrovirus. As with other viruses, it contains a protein coat, a capsid, surrounded by a phospholipid bilayer, which is formed from the host cell's cell membrane. Glycoproteins in that phospholipid bilayer allow the virus to reinfect new host cells. So how does HIV infect us, and why does it cause so much damage and ultimately may even lead to the death of some people? So HIV infects T lymphocytes. It binds to the CD4 receptor, which we've met previously, present on the outside of the T helper cell. The virus capsid fuses with the cell surface membrane, and RNA and reverse transcriptase enter those T helper cells. The reverse transcriptase enzyme is incredibly clever. It converts the viral RNA into DNA By doing so, the viral DNA is inserted into the host DNA, and viral mRNA is produced which can code for viral proteins. If you think about it, the HIV virus is terrifyingly clever. So how does HIV develop into AIDS, the disease? Well, as I've said, the HIV infects the T lymphocytes, which causes a reduction in active lymphocytes. If we have a reduction in active lymphocytes, it means that we produce fewer antibodies, which fundamentally lowers our immunity. And it means that infections, which usually wouldn't cause a massive problem for us, become far more lethal. So how is HIV spread? Well, it's spread through small cuts in the penis, mouth, vagina and intestine during sex. Infected blood transfusions. If you've got hypodermic drug users sharing needles, it's spread in that way. It can spread across the placenta when a woman is pregnant, and even through breastfeeding. When we think about the treatment options for HIV, why can't we use antibiotics? And that's because antibiotics can only be used to kill bacteria, and that's due to the composition of bacterial cells. Remember, they have that cell wall. Antibiotics work by causing that cell wall to be weakened, fundamentally meaning that the bacterial cell bursts open. Because viruses are non-living, they don't have a metabolism, they work in a very different way to bacterial cells. They don't have cell walls. Remember, they have capsids and proteins. They therefore cannot be affected by antibiotics. Describe how the human immunodeficiency virus is replicated once inside helper T cells. So it says very clearly that we need to talk about what happens once that HIV virus is inside the T helper cell. So it's that enzyme reverse transcriptase, which, remember, converts that viral RNA to DNA in the host nucleus, meaning that that viral DNA is incorporated to the host DNA. So unfortunately for that host cell, that host T helper cell, the viral mRNA produced is then able to code for more viral proteins. HIV-1 is the most common type of HIV. HIV-1 binds to the receptors on helper T cells called CCR5. Current treatment for HIV-1 involves the use of daily antiretroviral therapy, ART, to stop the virus being replicated. Only 59% of HIV-positive individuals have access to ART. Scientists have found that 2 HIV-1 positive patients, P and Q, have gone into remission. They have no detectable HIV-1. This happened after a blood stem cell transplant, BSCT. Patient P was given 2 BSCTs, Q was given 1 BSCT. All BSCTs came from a donor with helper T cells without the CCR5 receptor. In addition, patient P had radiotherapy and patient Q had chemotherapy. Both of these treatments are toxic. Both patients stopped receiving ART 16 months after BSCT. 18 months after stopping ART, both patients had no HIV-1 RNA in their plasma, no HIV-1 DNA in their T helper cells, and no CCR5 on their T helper cells. Use information given to evaluate the use of BST to treat HIV infections. Remember, when you're asked to evaluate, you'll give arguments for and against whatever statement they've made, and you always want to comment on the quality of the study in terms of how many patients were looked at, how long term we think the conclusions drawn will be valid for. So arguments for, and you really can just write it like this, for the use of BSCT include the fact that after 18 months, no HIV-1 RNA is present, and let's make that comment, no CCR5 receptor is present on the TH cells, so there's nowhere for the HIV to bind. These are both examples of how BCST is acting as an effective cure. Now let's move on to arguments against, saying that we can't just necessarily agree with all these conclusions. Include the fact we have no way of knowing if chemotherapy is still needed. or if it's solely responsible for the good outcome. And now I'm going to squeeze in some comments to do with the actual study. Only 2 people were tested and only HIV-1 patients were tested. Explain how HIV affects the production of antibodies when AIDS develops in a person. That's clearly going to decrease the production of antibodies, but why does it do that? Think about the type of cell the HIV infects. So HIV destroys helper T cells, meaning fewer B cells are activated. Remember, that's the role of T helper cells, so fewer antibodies are produced. We're now going to turn our attention to monoclonal antibodies. As the name suggests, mono meaning one type, cloning meaning the production of many. So you're producing a large number of identical antibodies, and they have far-reaching uses within medicine. The reason why they're so useful is the antibodies are made which are specific to particular antigens. And as I've already said, these antibodies can be isolated and cloned, so we produce large numbers of these useful antibodies. Monoclonal antibodies can be used to treat cancer. That's because the monoclonal antibody is complementary to the antigens on cancer cells. They can be used to block chemical signals and therefore can help reduce uncontrolled growth and prevent the growth of tumours. So how are monoclonal antibodies produced? Well, you need to take lymphocytes because obviously these produce the all-important antibodies, but lymphocytes reproduce fairly slowly. So if you're thinking you need large numbers of antibodies, that's not going to be a particularly quick way of getting to that end result. However, what type of cells replicate super fast? Well, tumour cells. So in effect, what you need to do is combine your lymphocytes, which actually come from mice, they're mouse lymphocytes. You combine them with a tumour cell to produce what's known as a hybridoma. Hybrid meaning a combination. And the hybridoma produces large numbers of that specific monoclonal antibody. One example of the use of monoclonal antibodies is by preventing regulator proteins from working. How do they do that? Well, remember, because they're types of antibodies have variable regions which are complementary to the protein, so they bind to the regulatory protein, and therefore the regulator protein can no longer bind to its receptor in the cell. You can also use monoclonal antibodies in what's known as indirect monoclonal antibody therapy, and this is when a therapeutic drug is attached to the monoclonal antibody. Because that antibody is specific, it attaches to a particular antigen, it can therefore deliver the drug to that cell with pinpoint precision. Monoclonal antibodies can also be used in medical diagnosis. In this particular case, the monoclonal antibody is, as always, complementary to a particular antigen. Now, when that monoclonal antibody binds to that antigen, that in effect identifies the antigen, and therefore the cell that produces that antigen is being present in the body. One of the most famous examples of the use of monoclonal antibodies is in pregnancy tests, because it's used to show the presence of a pregnancy hormone known as HCG, human chorionic gonadotrophin. So how does this pregnancy test work, and what is the role of the monoclonal antibodies in it? Overall, the test works by detecting the presence of that HCG hormone in the urine. This hormone is so small it's able to pass through the glomerulus and enter the Bowman's capsule of the kidney nephron, and therefore pass all the way along that nephron to the urine. Now, mobile monoclonal antibodies are attached to a dye. The hormone binds to a complementary antibody. Monoclonal antibodies specific to the HCG antibody complex become immobilized on the pregnancy test stick, and the binding of the HCG antibody complex triggers the appearance of colour, so the line becomes visible. Now, there's a second line of antibodies which are complementary to the mobile antibodies only, and that the role of those is to just confirm that the test is working. It's acting as the control. So 2 lines in this case indicate that you're pregnant. Another very important use of monoclonal antibodies is in the ELISA test, which is used to test for HIV. So how does the ELISA test work? You fix the monoclonal antibody specific to the HIV antigen to wells in a spotting tile. You add your sample, and any specific HIV antigens present will bind to your monoclonal antibody. You then wash your wells in order to remove any unbound antigen. You add a second set of monoclonal antibodies, which are linked to colour-changing enzymes, and these bind to any antigens which you previously captured. You wash your wells again, and this removes any unbound detection antibodies. You then add a suitable or appropriate substrate, and this is going to trigger enzymes which catalyse a colour change. That colour change therefore represents the presence of the HIV antigen. They could ask you why the sample is washed twice, and that's in order to prevent false positive results. There are ethical concerns with the use of monoclonal antibodies. One of those is the fact that you're using mouse lymphocytes, so obviously there's major ethics with inducing tumour formation in mice. Monoclonal antibodies have been associated with some deaths during treatment, and some monoclonal antibodies cause severe side effects. ADCs are molecules made of a monoclonal antibody linked to a cancer drug. Figure 1 shows how an ADC enters and kills a tumour cell. The process of entering the cell and the breakdown of the antibody to release the drug is very similar to phagocytosis. So we have ADC binding to the antigen on the surface of our tumour cell. And look, this does look incredibly similar to phagocytosis. So when it gets enclosed and then is released, it's going to cause cell death. Use your knowledge of phagocytosis to describe how an ADC enters and kills the tumour cell. That's worth 3 marks. So really we're describing the steps involved in phagocytosis. So we're going to start by saying that the cell engulfs the ADC. Remember that this here is a lysosome, and that's going to fuse with our phagosome, which contains the ADC. The lysosome then secretes our hydrolytic enzymes, which cause the release of ADC into the cell. Some of the antigens found on the surface of tumour cells are also found on the surface of healthy human cells. Use this information to explain why treatment with an ADC often causes side effects. If ADC is causing cell death, then obviously it's going to cause side effects if it's killing our lovely healthy human cells. So we need to say that ADC will also bind to healthy cells and cause death or damage to them. Scientists investigated whether one type of ADC could be used to treat human breast cancer. This ADC is a monoclonal antibody combined with a drug to inhibit mitosis. The monoclonal antibody binds to a protein found on human breast cancer cells. That's good, because mitosis is a type of cell division. So if we bind our ADC to our tumour cells of our breast cancer cells, if we bind our ADC to human breast cancer cells, then this drug should therefore inhibit mitosis and therefore stop the uncontrolled cell division. The scientists placed small pieces of human breast cancer tissue under the skin of mice. The scientists then randomly divided the mice into 3 groups. They treated the groups as follows on day 0. Group G was our control. Group H was injected with monoclonal antibody. Group J was injected with ADC. So that's the monoclonal antibody plus the drug. Every few days, the scientists measured the volume of tumours formed from human breast cancer tissue. Figure 2 shows the scientists' results. So on the y-axis, we have the mean tumour volume, and on the x-axis, time after injection. You might as well label the key here. So that was control. This was monoclonal antibodies only, and this was the monoclonal antibodies plus the drug, ADC. Mice in group H were injected with 2 milligrams per kilogram of monoclonal antibody. The monoclonal antibody was in a solution of concentration of this. Calculate the volume of antibody solution that the scientists would have injected into a 23-gram mouse. Give your answer in DM cubed and standard form. The hardest thing with these questions is dealing with the units, so you must pay super careful attention to these. So it says the mice were injected with 2 milligrams per kilogram of monoclonal antibody, and we're told that we have a mouse which has a mass of 23 grams. So the first thing we want to do is find out the mass of the mouse in kilograms. So we just take that 23 grams and divide it by 1,000. Then looking more closely, so we know that they're injected with 2 milligram per kilogram. So we need to multiply that by 2 to find the mass injected into the mouse. And then lastly, we know we're looking for a volume of antibody solution. Our answer needs to be in decimeter cubed. Now we need to pay super close attention to our units. We know that the concentration of the monoclonal antibodies was in milligrams per decimeter cubed, so we need a way of cancelling out our masses in order to get, in effect, a volume. So I'll show you that now. It's easier if we look at the maths. So we take our 0.046, which was our mass of antibody injected into the mouse. We divide it by 500, which we know was milligram per decimeter cubed. This enables our milligrams to cancel out. So you're left with a unit which is 1 over decimeter to the minus 3, which is the same as decimeters cubed, which is great because after all that is a unit of volume. I do agree this is a lot of work though for 2 marks. And we're in standard form and we've got our volume unit in decimeters cubed. Give one reason why there were no data for Group G and Group H after day 8. If we check the information in the question, it tells us that the Group G were the control mice that had simply the human breast cancer tissue under their skin. Group H had just the monoclonal antibody as well as that breast cancer tissue, whereas Group J had the ADC, which is the drug. So the reason why there's no data after day 8, sadly, is likely because all the mice died. The mice died for those 2 groups of mice which hadn't received the ADC. Suggest and explain 2 further investigations that should be done before this ADC is tested on human breast cancer patients. So this is all to do with drug development during preclinical and clinical trials. So obviously when we develop new drugs, we need to test on healthy humans as opposed to ones with breast cancer to check for efficacy, so how well it works, check for any side effects. And we also need to investigate different concentrations of ADC to find the suitable dose.